Gametogenesis refers to the process of the development of haploid gametes from diploid germ cells. In animals, gametogenesis occurs in specialized organs such as ovaries in females and testes in males. In females, meiosis occurs in the ovary to produce haploid cells, while in males, meiosis occurs in the testes to produce haploid sperm. Gametogenesis is a crucial process that ensures genetic diversity and transmission of genetic information to the next generation. Fertilization is the process by which haploid gametes fuse to form a diploid zygote. In animals, fertilization occurs externally or internally depending on the species. In external fertilization, the sperm and egg unite outside the female's body, whereas in internal fertilization, the sperm are deposited inside the female's body where they fertilize the egg. Fertilization is followed by the formation of a zygote, which is the first step in the development of a new organism. Early development refers to the initial stages of embryonic development, which include during gastrulation, the blastula undergoes a complex series of movements to form the germ layers ectoderm, endoderm, and mesoderm. The ectoderm gives rise to the skin, nerve system, and other tissues, and the endoderm gives rise to the digestive system and respiratory system, and mesoderm gives rise to muscle, bone, and other tissues. Germ layers are unique to animals and represent the basic body plan of animals. In plants, embryogenesis occurs in the ovule, which is a part of flower. After fertilization, the zygote undergoes several rounds of cell division to form the embryo. The embryo then develops into a seed, which contains a dormant plant embryo and a food source. When conditions are favorable, the seed germinates and the embryo develops to a mature plant. Then morphogenesis and organogenesis and related topics. Morphogenesis is the process of forming the shape and structure of an organism, while organogenesis refers to the development of organs from the germ layers during embryonic development. In animals, this involves various processes such as cell aggregation, differentiation, patterning, and growth. Here are the detailed explanation of these processes. Cell aggregation and differentiation. Dictyostelium. Dictyostelium is a model organism used to study morphogenesis. During development, individual cells aggregate to form multicellular structures which then differentiate into various cell types. This process involves cell-to-cell communication and the activation of specific genes that regulate cell fate determination, then axis and pattern formation in Drosophila, amphibians, and chick. This organism have distinct axes anterior, posterior, dorsal, ventral, and left, right that are established during early embryonic development. The formation of this axis is controlled by maternal effect genes that are located in specific regions of the egg. Later, pattern formation occurs through the activation of signaling pathways that regulate the expression of genes involved in cell fate determination. Then organogenesis, vulva formation in Caenorhabditis elegans, islands, induction, limb development, and regeneration in vertebrates. During organogenesis, the germ layers differentiate into specific organs. In Caenorhabditis elegans, the vulva is formed from a subset of cells that undergo a series of cell fate decisions. Islands induction involves the formation of a lens placode that invaginates into form a lens vesicle which then differentiate into the lungs. Limb development and regeneration involve the activation of specific signaling pathways that regulate the proliferation and differentiation of progenitor cells. Then differentiation of neurons and related topics. So differentiation of neurons, post-embryonic development, novel formation, metamorphosis, neuronal differentiation involves the activation of specific genes that regulate the differentiation of precursor cells into neurons. Post-embryonic development involves the formation of different larval stages that undergo metamorphosis to form the adult form. This process involves extensive remodeling of the body plan and the activation of specific genes that regulate cell fate determination. Environmental regulation of normal development. Environmental factors such as temperature, light, and nutrient availability can have a significant impact on the normal development of an organism. For example, the sex of some reptiles is determined by the temperature at which eggs are incubated. Sex determination in animals, sex determination can be genetic or environmental. Genetic sex determination involves the presence of specific sex chromosomes, while environmental sex determination involves the influence of environmental factors. Understanding the process of morphogenesis and organogenesis is crucial for understanding normal development as well as developmental disorders and diseases. Studying model organisms has helped us to unravel many of the underlying mechanisms of this process, which then be applied to other mechanisms, including humans. Then, morphogenesis and organogenesis in plants. So, morphogenesis and organogenesis in plants refer to the development of various plant structures and organs from simple embryonic tissues. It involves the coordination of various developmental processes such as cell differentiation, cell division, cell expansion, and programmed cell death. The process is regulated by genetic, epigenetic, and environmental factors. In this context, the following are the major concepts related to the morphogenesis and organogenesis in plants. Organization of shoot and root apical meristem. Apical meristem is a region of active cell division and differentiation present on the tips of the shoot and root. It gives rise to all the above ground and below ground organs respectively. The organization of the apical meristem is a result of the interactions between different genes, hormones, and mechanical forces. Shoot and root development. The development of the shoot and root involves the formation of various organs such as leaves, stems, branches, and lateral roots. These processes are regulated by complex genetic networks involving the interaction of various signaling pathways such as auxin, cytokinin, gibberellins, and abscisic acid. Leaf development and phyllotaxy. Leaf development involves the differentiation of cells into different tissues such as epidermis, mesophyll, and vascular tissue. Phyllotaxy refers to the arrangement of leaves on the stem, and it is regulated by the activity of various genes and hormones. Then transition to flowering. Floral meristems and floral development. The transition to flowering is a crucial event in plant development, and it is regulated by various endogenous and environmental factors. It involves the conversion of vegetative meristem to the floral meristem, which gives rise to reproductive organs such as the sepals, petals, stamens, and carpels. The development of these floral organs is regulated by a complex genetic network involving various signaling pathways such as the photoperiodic pathway, the vernalization pathway, and the autonomous pathway. In Arabidopsis and Antherium, the molecular mechanisms underlying the floral development have been extensively studied. The floral meristem identity genes such as APETALA1, AP1, and leafy LFY are the key regulators of floral development. The expression of these genes is regulated by various pathways including the photoperiodic pathway and the autonomous pathway. The floral organ identify genes such as AFETALA3, AP3, and Pistillata PI, Agamus AG, and Sepalata SEP are also essential for the development of special floral organs. Overall, the morphogenesis and organogenesis in plants involve complex molecular mechanisms and interactions among various genes, hormones, and environmental factors. Understanding these processes is crucial for improving crop yields, plant breeding, and developing new varieties of plants with desired characteristics. Then programmed cell death, aging, and quiescence, each in detail. So, programmed cell death, aging, and senescence are important biological processes that occur in both plants and animals. Programmed cell death, also known as apoptosis, is the controlled destruction of a cell by the activation of specific intracellular processes. It is an essential process for the normal development and maintenance of tissues and organs, and it helps to remove damaged, infected, or unwanted cells from the body. The key features of programmed cell death include the activation of specific genes, the fragmentation of DNA, and the removal of cellular debris by phagocytic cells. Examples of programmed cell death include the removal of webbing between fingers and toes during embryonic development, the elimination of damaged cells in response to radiation or chemotherapy, and the elimination of infected cells during an immune response. Aging is the progressive decline in physiological function that occurs with advanced age. This is a complex process that involves a wide range of cellular and molecular changes, including the accumulation of DNA damage, telomere shortening, oxidative stress, and inflammation. These changes can lead to a decline in tissue function and increased susceptibility to disease and ultimately death. Aging is a natural process that occurs in all organisms, and it is influenced by both genetic and environmental factors. Some examples of age-related changes include the decline in muscle mass and strength, an decrease in cognitive function, and an increased risk of chronic diseases such as cancer and heart disease. Senescence is a state of irreversible growth arrest that occurs in cells in response to various stresses, including DNA damage, telomere shortening, and oncogene activation. It is a protective mechanism that helps to prevent growth of damaged or potentially cancerous cells. The key features of senescence include the activation of specific genes, the production of inflammatory cytokines, and the secretion of extracellular matrix degrading enzymes. Senescent cells are known to accumulate with age and contribute to age-related diseases and tissue dysfunction. Examples of senescence include the replicative senescence of cultured cells, the senescence of skin cells in response to UV radiation, and the senescence of immune cells during aging. System physiology, plant, photosynthesis, and all related pathways. Photosynthesis is a fundamental process by which plants convert light energy into chemical energy, resulting in the production of carbohydrates, oxygen, and water. The process of photosynthesis can be divided into two main stages: light dependent reaction and light independent reaction. Light dependent reaction occur in the thylakoid membrane of the chloroplast and involve the absorption of light by photosynthetic pigments such as chlorophyll, which is found in photosystems 1 and 2. The absorbed energy is used to generate ATP and NADPH, which are used as energy carriers in the light-independent reactions. In addition to generating ATP and NADPH, the light-dependent reactions can also produce harmful reactive oxygen species, ROS, and byproducts. Plants have developed photoprotective mechanisms to prevent damage from this ROS, such as the xanthophyll cycle, which involves the conversion of vioxanthin to zeaxanthin in response to excess light. The light-independent reactions occur in the stroma of the chloroplasts and involve the fixation of CO2 to produce carbohydrates. The most common pathway of CO2 fixation is the C3 pathway, which occurs in the majority of plants. In this pathway, CO2 is fixed by the enzyme ribulose biphosphate carboxylase oxygenase or Rubisco to produce a 3-carbon compound. However, the C3 pathway can be inefficient in certain environmental conditions such as high temperatures or water deficits. Some plants have evolved alternative pathways such as the C4 and CAM pathway, which are more efficient in this modifications. In the C4 pathway, CO2 is first fixed into a 4-carbon compound in mesophyll cells before being transported to bundle sheath cells where it is decarboxylated and fixed by Rubisco. This pathway is found in plants such as maize and sugarcane. In the CAM pathway, CO2 is fixed into a 4-carbon compound at night when stoma are opened and then stored until the day when it is decarboxylated and fixed by Rubisco. This pathway is found in plants such as cacti and succulents. Overall, the process of photosynthesis is essential for the survival of plants and is an important source of food and oxygen for other organisms. The different pathways of CO2 fixation have allowed plants to adapt to a variety of environmental conditions, highlighting the importance of understanding the physiology of plants in the context of their environment. Respiration and photorespiration, citric acid cycle, plant, mitochondrial, electrical transport, and ATP synthesis, and all those related topics. Respiration is the process by which living organisms release energy from organic compounds. In plants, respiration occurs in the mitochondria and involves the breakdown of glucose to produce ATP, which is used for various cellular activities. Then citric acid cycle. The citric acid cycle, also known as the Krebs cycle or tricarboxylic acid cycle, is a series of reactions that occur in the mitochondria of plant cells. It involves the breakdown of acetyl-CoA to produce NADH and FADH2, which carry electrons to the electron transport chain for ATP synthesis. Plant mitochondrial electron transport and ATP synthesis. The electron transport chain is a series of complexes that transfer electrons from NADH and FADH2 to oxygen, producing ATP through oxidative phosphorylation. In plants, the electron transport chain occurs in the mitochondria. Then alternative oxidase. The alternative oxidase is an enzyme found in the mitochondria of some plants that bypasses some of the complexes of the electron transport chain, leading to the production of heat instead of ATP. This process is called alternative respiration and can be important for thermogenesis in certain plants. Photorespiratory pathway. Photorespiration is a process that occurs in the chloroplasts of plants and involves the uptake of oxygen and release of carbon dioxide. This process can be wasteful for plants because it reduces the efficiency of photosynthesis. However, plants have developed mechanisms to reduce photorespiration through C4 plants and CAM pathways. Then nitrogen metabolism and related topics. Nitrogen is an essential macronutrient required for plant growth and development. It is required for the synthesis of amino acids, nucleotides, and other important molecules. However, nitrogen is often limiting in many soils, so plants have evolved mechanisms to efficiently acquire and utilize nitrogen. Nitrate and ammonium are the two major forms of nitrogen that plants can absorb from the soil. Nitrate is the most abundant form of nitrogen in aerobic soils, while ammonium is more prevalent in anaerobic soils. Plants use transporter to take up these nitrogen sources and then convert them to organic forms through the process of assimilation. Nitrate assimilation involves the reduction of nitrate to ammonium, which is then incorporated into amino acids and other nitrogen containing compounds. This process occurs in the cytosol of root cells and leaves. Ammonium assimilation involves the direct incorporation of ammonium into organic compounds such as glutamate and glutamine, which serve as precursors for other amino acids. Amino acids are the building blocks of proteins and are essential for many physiological processes in plants. Plants can synthesize all 20 amino acids, but some are considered essential because they cannot be synthesized in sufficient quantities to meet the plant's needs. These essential amino acids must be obtained from the plant's environment. The biosynthesis of amino acids is a complex process that involves multiple enzymatic reactions. Each amino acid has a unique biosynthetic pathway that often involves the conversion of intermediates from other pathways. For example, the biosynthesis of serine involves the conversion of 3-phosphoglycerate, an intermediate of glycolysis, to serine. Similarly, the biosynthesis of tyrosine involves the conversion of phenylalanine, an essential amino acid. Overall, nitrogen metabolism is essential for plant growth and development. Efficient nitrogen assimilation and amino acid biosynthesis are critical for producing healthy, high-yielding crops. Understanding the mechanisms of nitrogen metabolism in plants can help improve agricultural practices and enhance food security. Next, plant hormones. Plant hormones, also known as phytohormones, are organic compounds that regulate various physiological processes in plants. There are 5 major types of plant hormones: auxins, cytokines, gibberellins, abscisic acid, and ethylene. Each of these hormones plays a unique role in plant growth and development. Auxins are responsible for promoting cell elongation and cell division, as well as regulating the direction of growth in response to light and activity. Indole 3-acetic acid, or IAA, is the most common and important auxin found in plants. Auxins are also involved in root development, apical dominance, and fruit development. Then cytokines. Cytokines are involved in cell division, shoot and root growth, and the delay of senescence. They are produced in the root tips and transported upward to the shoot. The cytokinin to auxin ratio determines the rate of cell division and differentiation. Then gibberellins promote stem elongation, seed germination, and fruit development. They also play a key role in leaf expansion and flower development. Gibberellic acid is the most commonly known as and studied gibberellin. Abscisic acid. Abscisic acid is involved in seed dormancy, drought tolerance, and the inhibition of seed germination. It also regulates stomatal closure in response to water stress. Ethylene. Ethylene is a gas hormone that is involved in fruit ripening and leaf senescence and the response to mechanical stress. It also regulates cell division and the differentiation in the root and is involved in the response to pathogens. Plant hormones are synthesized in various parts of the plant, including the shoot, apex, roots, and developing seeds. They are transported to other parts of the plant through the phloem and xylem. Plant hormones bind to specific receptors on the cell surface or within the cell, leading to a signal transduction pathway that regulates gene expression and other physiological processes. The importance of plant hormones lies in their ability to regulate plant growth and development in response to various environmental cues such as light, temperature, and water availability. They also play a crucial role in the response of biotic and abiotic stress such as pathogen attack and drought. Understanding the mechanisms of plant hormone action is important for developing new strategies for crop improvement and plant protection. Then sensory physiobiology, photobiology, and related topic. Sensory photobiology is the study of how plants perceive and respond to light signals. Here are some key topics related to this field. Phytochromes. These are the light receptors that are sensitive to red and far red light. They regulate many developmental processes including seed germination, stem elongation, and flowering. Phytochromes exist in two forms: PR, red light absorbing, and PFR, far red light absorbing. Then when PR absorbs red light, it is converted into PFR. Conversely, when PFR absorbs far red light, it is converted back into PR. This reversible conversion allows plants to monitor the light environment and adjust their growth and development accordingly. Then cryptochromes. These are blue light receptors that regulate many physiological processes including circadian rhythms, photomorphogenesis, and shade avoidance. Cryptochromes are involved in both the perception and response to light signals. They interact with various signaling pathways including the photoperiodic pathway and circadian clock and the auxin signaling pathway. Then phototropins. These are blue light receptors that control phototropism, the directional growth of plants in response to light. Phototropins are involved in many other processes as well, including stomatal movement, chloroplast movement, and photomorphogenesis. They interact with a variety of signaling pathways, including auxin signaling pathway and the cytoskeleton. Then stomatal movement. Stomata pose on the surface of leaves that regulate gas exchange and water loss. Stomata open in response to light, allowing the plant to take up carbon dioxide for photosynthesis. Stomatal opening is regulated by a complex interplay of environmental and hormonal signals including light, CO2, abscisic acid, and cytokines. Photoperiodism. This is the physiological response of plants to the length of day and night. Photoperiodism regulates many developmental processes including flowering, tuberization, and dormancy. The photoperiodic pathway involves a complex network of signaling molecules including phytochromes, cryptochromes, and the circadian clock. Then biological clock. These are endogenous rhythms that allow plants to anticipate and adapt to environmental changes. Biological clocks regulate many physiological processes including growth, development, and metabolism. The circadian clock is a central regulator of biological rhythms in plants and it is ultimately linked with the photoperiodic pathway. Then solute transport and photoassimilate uptake and all those related topics. So solute transport and photoassimilate translocation are vital processes in plant physiology that ensure the proper uptake, distribution, and utilization of nutrients for growth and development. Here are the major important points and examples for each process. Solute transport. Solute transport involves the uptake and movement of water, ions, and other solutes from the soil into the plant roots, through the cells, and across the membranes. The movement of solutes is driven by gradients in concentration and electric potential. There are two major pathways for solute transport: the apoplastic pathway, movement through cell walls, and the symplastic pathway, movement through plasmodesmata. The transport of water and nutrients is tightly regulated to maintain proper balance and prevent excessive uptake or loss. Examples of solute transport mechanisms include ion channels, transporters, and pumps. Then photoassimilate translocation. Photoassimilate translocation involves the transport of organic compounds such as sugars, amino acids, and hormones from the source tissue where they are synthesized to the sink tissues where they are utilized or stored. The main pathway for photoassimilate translocation is the phloem, which is composed of specialized sieve tube elements and companion cells. Photoassimilates are loaded into the phloem at the source tissue and unloaded at the sink tissue by active transport mechanisms. The movement of photoassimilates is driven by gradients in concentration and tissue. Examples of photoassimilated transport mechanisms include sucrose transporters and H plus sucrose symporters. Importance: solute transport and photoassimilate translocation are essential for plant growth and development and reproduction. This process ensures the proper uptake and utilization of nutrients for various physiological processes such as photosynthesis, respiration, and biosynthesis. They also play important roles in plant response to environmental stresses such as drought, salinity, and nutrient deficiency. Understanding these mechanisms of solute transport and photoassimilate translocation is essential for developing strategies to enhance crop yield and quality, as well as for improving plant adaptation to changing environmental conditions. So next topic, secondary metabolites and related topics. Plants produce a diverse array of secondary metabolites which are not directly involved in growth, development, or reproduction but play important ecological roles such as defense against herbivores, pathogens, and environmental stresses, attraction of pollinators, and competition for resources. Some important secondary metabolites are terpenes, phenols, and nitrogenous compounds. Terpenes are a class of compounds derived from 5-carbon isoprene units. They play a critical role in plant defense against herbivores and pathogens, as well as in attraction of pollinators and seed dispersers. Terpenes also have medicinal properties and are used in the production of fragrances, flavors, and pharmaceuticals. Examples of terpenes include menthol, linolene, and carotenoids. Phenols are another class of secondary metabolites which play a role in plant defense against pathogens and environmental stresses. They are also involved in regulating plant growth and development. Examples of phenols include lignin, tannin, and flavonoids. Flavonoids are a diverse group of phenolic compounds that are responsible for the pigmentation of flowers and fruits and are also important in plant defenses against UV radiation and insect herbivores. Next, nitrogenous compounds such as alkaloids, cyanogenic glucosides, and glucosinolates are also important secondary metabolites in plants. Alkaloids are nitrogen containing compounds that have diverse biological activities including defense against herbivore, attraction of pollinators, and antimicrobial properties. Cyanogenic glycosides are compounds that release hydrogen cyanide upon hydrolysis, which serves as a potent deterrent against herbivores. Glucosinolates are sulfur-containing compounds that play a role in plant defense against insect herbivores and pathogens. The biosynthesis of secondary metabolites is regulated by various environmental and developmental signals such as light, temperature, and stress, as well as by plant hormones. The diversity of secondary metabolites in plants is vast, and their importance in various ecological, biological, medicinal, and industrial applications cannot be overstated. Secondary metabolites are organic compounds that are not essential for the survival of the plant but play often very important roles related with the plant's interaction with the environment. Terpenes, phenols, and nitrogenous compounds are examples of secondary metabolites in plants. Terpenes are a large and diverse class of compounds that are synthesized from isoprene units. They play important roles in plant defense against herbivores and pathogens, as well as in attraction of pollinators and seed dispersers. Examples of terpenes include essential oils found in many plants such as mint, lavender, and citrus fruits. Phenols are a class of aromatic compounds that are synthesized from phenylalanine or tyrosine. They are involved in plant defenses against pathogens and herbivores, as well as in UV protection and regulation of growth and development. Examples of phenols include flavonoids, which are responsible for the coloration of flowers and fruits, and tannins, which are found in many woody plants and are involved in defense against herbivores. Next topic we will be learning tomorrow.